This episode of the Curious About Cannabis podcast is brought to you by you, our dedicated listeners and supporters. Thanks to your continued listening, sharing, and donations, we've been able to continue the show free from third-party advertisers and sponsors. So, thank you. And if you'd like to learn about other ways you can support the show, visit patreon.com/curiousaboutcannabis.
My name is Vincenzo Di Marzo. I'm the research director at the National Research Council, and currently I'm actually a Canada Excellence Research Chair in Quebec City in Canada. I've been working on the endocannabinoid system since basically its discovery at the beginning of the 1990s, and I've been working on the biochemistry, chemistry, pharmacology, development of new tools, analytical tools, pharmacological tools to investigate this system. And more recently, I've become interested in investigating the expansion of the endocannabinoid system into a larger chemical signaling system, which we define as the endocannabinoidome, and the relationship between the endocannabinoidome and the gut microbiome, which is what I do in Quebec City at Université Laval.
You're listening to the Curious About Cannabis podcast. Hey everybody, this is Jason Wilson with the Curious About Cannabis podcast. Thanks so much for tuning in once again. So today I am beyond delighted to connect with somebody I've been wanting to talk to for many years since I started studying cannabis and cannabinoid science. I'm with Dr. Vincenzo Di Marzo, who, if you haven't heard of him, by the time you finish listening to most of my podcast episodes, you will definitely have heard of him. Thanks so much, Dr. Di Marzo, for being willing to come on the podcast today and talk to me about cannabinoid science.
My pleasure. Thank you for inviting me.
Yes, absolutely. So one of my first questions, in the beginning of your book, The Endocannabinoidome, in your dedications, one thing that you admit is you have an obsession about endocannabinoids and cannabinoids. So I guess my first question to you is, why are you so obsessed with cannabinoids?
Well, I would say more than cannabinoids, I'm obsessed with the endogenous counterparts of the cannabinoids, which are the endocannabinoids, and in fact, since the beginning, when we started investigating this chemical signaling system, we were intrigued and fascinated by its involvement in basically all physiological aspects of mammalian life. And then we also understood that, in fact, this is a system so deeply involved in physiology that becomes also deeply involved in pathology. And it seemed to be quite complicated to study, but then it became even more complicated when we could see that this system was not as simple as we thought at the beginning, but it was easily to be expanded into a much bigger signaling systems, which probably we will end up discovering controls all aspects of human physiology and pathology. And the other reason why I became so interested in this system is because there is a lot of space for chemistry. I'm a chemist to start with, and there is a lot of chemical variability and biodiversity and chemical diversity in not only the cannabis plant but also in the endogenous signals that we discovered starting from studies on THC. So the more we studied it, the more complicated it looked. So I really like complicated things. I like things that can explain in a mechanistic way things from the functioning of a single cell up to the behavior of a complex organism like laboratory animals and humans. So I still think that we've probably done less than 10% or 15%, and one lifetime will not be enough to study it all.
Yeah, absolutely. Well, and that just gives plenty of work to the future generations. Something that's been on my mind a lot. That I was emailing Raphael Meshulam just a couple of days ago and asking him about I don't know what he hopes that people take away from the work that himself and you and other people like Roger Pertwee and others who are really kind of
Yeah.
at the beginning of a lot of the stuff, where it hopes to go. And we were talking a little bit about the future generations of cannabinoid scientists that are coming on board with new opportunities to study cannabinoids that is much more difficult back in through the 60s to the 90s or so, when we're really getting a lot of this grounding established. It's a really exciting time to see what the subsequent lifetimes
Yeah.
unveil that we won't be around to see, and how
I
that
was
influences
very
medicine.
lucky to start in this field when the pioneers of this field, like Reframing and Roger Pertwee and Alan Howlett, were really still very, very active in this field. They're still active, of course, but obviously those were the times when they were making also themselves the major discoveries in this field, and this also explains my excitement at being there. And I've been very lucky because I when I think about my involvement in this field, I thought I was in the right place at the right time. So
Yes.
I owe a lot of what we did in my group to the work of Rafi and Roger and Alan Holt.
And how, so talking about your start in doing some of this research, um, how did you start to get clued into? I mean, obviously you were involved in foundational research that characterized the concept of the entourage effect, and as well as your paper in 1998 that sort of described for the first time what the endocannabinoid system does and that sort of thing. But start to when did the idea of the endocannabinoid system start to expand for you? When did you start to think about? It seems like there was a perceptual shift in how we saw fatty acids and all these other
Yeah.
components of the body once we started studying these cannabinoid receptors and how the vanilloid receptors are part of all these things. So when did that start to really expand for you, where you realized this is something way bigger than
Mhm.
just CB1, CB2, anandamide, 2-AG, all of that?
Well, yeah, that's a difficult question, but
Yeah.
obviously the realization of the importance of the endocannabinoid system came, I would say, between, in my case, at least 1995 and 2000, because at those times we actually developed analytical methods to measure the endocannabinoids and anandamide and 2-AG in biological fluids and tissues. And we were very, very lucky to collaborate with tens, I would say even probably hundreds of scientists all over the world who would provide us with samples from patients with various disorders or from animal models of disorders. So we basically started analyzing tissues and blood like meds all over the world, and we realized that this system was very easily perturbed by
Mm.
several different, not only pathological conditions, but also um, changes in the equilibrium, in the physiological equilibrium. Even physiological changes would change the endocannabinoid system. So I and others came to the conviction that this system is a pro-homeostatic system, uh,
Mm.
present in all cells and tissues. And especially if you expand this system beyond CB1 and CB2, you will find endocannabinoids like anandamide and 2-AG in all tissues from not only from mammals but also from higher vertebrates, and in some cases also from vertebrates. So we noticed, we started working also in some very primitive forms of life, like a hydra or some mollusks or echinoderms, and we could see that the endocannabinoids were there, even though the cannabinoid receptors, as we knew them, CB1 and CB2, were not there. So we wondered why do we make these molecules, and we also realized that the amounts of these molecules were subject to be altered by the diet
Mm.
and by the intake of some particular fatty acids in the diet. So we wondered how come that there is such an important system, and this system can be changed by something as simple as eating one oil rather than another. So we were really intrigued, and we thought that this maybe was really the response of the organism, if not the only one, but one of the most important responses of the organism to all forms of going far from the equilibrium, from the homeostasis, and this system would be activated to bring back homeostasis in cells and also at the organismal levels. And then we realized that several forms of stressful conditions would also alter this system in a way that this system could change the output of other chemical signals. So in those years, we came to the hypothesis, and it was still an hypothesis that has been since then, I would say, largely proven, that the system is a system made of local chemical signals, because the endocannabinoids cannot really travel from one organ to the other, so they act where they are produced. Produced and triggered by stressful, even kind of benign forms of stress, subtle perturbation of the status of the cell, but also environmental stress, like physical exercise, diet, changes in the light-dark cycles, in order to retune the output of other chemical signals. But in turn, since this system was present in different parts of the organism, the system was also itself being controlled by many other chemical signals. So it's like we made a comparison. It's like an orchestra. It's like the director of the orchestra, which is also directed by the musicians of the orchestra. So it was quite an intriguing hypothesis that we're still investigating. And of course, now with the discovery of the endocannabinoidome, this is becoming more difficult to prove.
Yes, yeah, absolutely. And for those listening that maybe don't have such a technical background on cannabinoid science, how would you describe the concept of the endocannabinoidome to somebody?
Okay, so basically, with the word "ome", now we make new words containing the prefix, the suffix "ome", to identify a very large system. So we have the genome, we have the metabolome, we have the proteome, and so on and so forth. So the endocannabinoidome came in a way from, you mentioned before the study that Rafi Meshulam, in which we collaborated on the entourage effect. So basically, what Rafi perceived is that, and what we were measuring, in fact, when we were measuring endocannabinoids, is that these two molecules, anandamide and 2-HG, were accompanied by a plethora of other compounds. And these compounds, these metabolites, were chemically and biochemically similar, meaning that anandamide and 2-HG belong to two different families of metabolites, and these are the N-acyl ethanolamines and the monoacyl glycerols, some of which were already known, in fact, even 1 or 2 decades, or even longer than that, before the endocannabinoids were discovered. So, but nobody knew really how these molecules were acting. So Rafi, in a very imaginative way, that this is really his main feature, is creativity and imagination, thought, well, these are accompanying molecules that are there to protect, to make the two big actors look even more important than they are. So it's an entourage, or when politicians go around and they take with them a lot of accompanying people, and that's certainly part of the picture. But then we started realizing that these compounds, in fact, do have molecules, do have targets of their own. So the endocannabinoidome, if you wish, is a kind of evolution of the entourage effect. Molecules like the N-acyl ethanolamines or the monoacyl glycerol started being investigated. Receptors were being found, and this created, of course, excitement but also complications.
Yes.
because the pharmacologists and the drug developers were the therapeutic drug developers were thinking, okay, we can manipulate the levels of since they are so strongly involved in pathology, we can manipulate the levels of the endocannabinoids of anandamide and 2-HG in order to make more of these compounds where they're needed, or to reduce their amounts where they're produced in an excessive manner. So you can do that by manipulating the degradation or the biosynthesis of endocannabinoids. But then when it was found that, and we and many others, that anandamide has the same biochemical pathways as other N-acetylamines, which have different receptors from the cannabinoid receptors. This created a strong complication. We cannot really manipulate the levels of anandamide in this way without manipulating the levels of the other previously known as entourage compounds, but having an identity and the mechanism of action of independent from the endocannabinoids of their own. So the endocannabinoidome is basically the bigger system, including not only anandamide and 2-HG and their main receptors, which are CB1, CB2, but also some of the other receptors for anandamide and 2-HG, because these are two very promiscuous molecules, unlike THC. This is one major difference between the endocannabinoids and the plant cannabinoids, or at least between the endocannabinoids and THC. And then it was found that, in fact, even the biosynthesis and degradation of these two compounds could not be only mediated by one or two enzymes. There was a redundancy of biosynthetic and degrading enzymes. And sometimes the degrading enzymes were not degrading enzymes because they were simply converting anandamide and 2-HG into molecules which had different receptors. And this may seem strange, but not to people like me, who have investigated bioactive lipids all their life. All bioactive lipids are transformed into something different and come from something different with a different biological activity. So, and then there is the congeners of anandamide and 2-HG, what we called previously the entourage compounds, which have their own receptors, even though they share with the endocannabinoids the same biosynthetical pathways. And then finally, there were other molecules which have different biochemical pathways as the endocannabinoids, but are nevertheless, they're not congeners, they're not like cousins, they're not like brothers and sisters, but they're cousins. So you have other N-acyl amides, and in fact, when we collaborated with the other groups, we found that, in fact, in particular with the group of Michael Walker, the late Michael Walker, we found that many amino acids could make amides with fatty acids, and this could create an almost an incredible number of new combinations between 10 or 15 fatty acids and 20 amino acids. So just imagine all the permutations. And and so these compounds actually exist, as the group of Heather Bradshaw has also recently investigated. And so it's really, we're talking now about a huge number of chemical mediators. So the endocannabinoids, their congeners, their analogs, which may or may have not different similar biochemical pathways, normally have different receptors. We talking about the several other targets that anandamide and 2-HG have beyond CB1 and CB2. We're talking about other enzymes. So we're talking about something about more than 250 chemical mediators, more than 50 enzymes and receptors. And some of these receptors were actually known previously, like the TRP channels or the PPARs. And the interesting thing is that, of course, when the endocannabinoid system became so big, then the next question was, okay, THC is the only out of 100 and more plant cannabinoid that interacts with the endocannabinoid system. What about maybe if we don't look only at the endocannabinoid system, but we look at the expanded endocannabinoid system, at this endocannabinoid, maybe the other cannabinoids will interact with the endocannabinoidome, even though they don't interact with the endocannabinoid system. This is true to a large extent for things like cannabidiol or cannabigerol, the other minor, not so minor anymore, cannabinoids, and in fact, in the case of cannabidiol, even the endocannabinoidome is not sufficient to explain its pharmacology, because it has been suggested to interact also with receptors and proteins outside the endocannabinoidome, not just outside the endocannabinoid system, but even outside the endocannabinoidome. So I think the endocannabinoidome is a complex concept, which, however, has the merit of explaining the mechanism of action of a much higher number of plant cannabinoids than the endocannabinoid system.
Yeah, absolutely. And it's definitely a very tangled web that you can't, so many things are interconnected that it is tricky to manipulate one piece. I saw, wasn't there research? I can't remember what year it was now. There was research, I think, in France, where they tried to do, I think it was a FAH inhibitory study, and ran into some problems there, unexpected consequences and things.
Yeah, even though in that particular case, the problem was not due to the fact that by inhibiting the degradation of anandamide, they were also inhibiting the degradation of other fatty acid amides. They basically were hitting with this new chemical. They were hitting a completely different set of proteins and enzymes for which there is less redundancy in the brain. So when you get rid of that enzyme, you have serious problems. Fortunately, when you inhibit Fa, you have much less side effects, because, as I mentioned, for the degradation of the endocannabinoids, there are many enzymes. So this is a system so strongly and deeply involved in mammalian and animal physiology that it's not enough to kick out one of the enzymes of the receptors to see major problems.
Right. Yeah. And one thing that's come up in several discussions I've had, both with Raphael Mechoulam and another colleague of yours, Ethan Russo, was how do we, I guess, taking this to a practical level, because what physicians are interested in is, okay, this is all interesting. We see it's connected to all of these aspects of physiology, but how do we measure it when these compounds are so kind of ephemeral? They get built when they're needed, and then they get broken down quickly. And I know that in the past there have been like spinal taps and stuff like that to try to see what endocannabinoids are circulating at the moment. But can you speak at all to the challenges to trying to characterize the endocannabinoid system or the endocannabinoidome in a human being?
Yeah, it's you're perfectly right. It's very complicated. It's a system that undergoes the levels of these mediators undergo changes by the hour. As I said, they're affected by whether or not you eat food or what kind of food you eat. Like with all the other ohms, the real picture of the system can only be taken at a certain time frame, thanks to the very sophisticated new machines that have been really have characterized the more recent years of scientific development. As you know, the means by which we can look into the cell, into the biological fluids, have become very sophisticated and very sensitive and very specific. So we can now measure the levels of anandamide, and we can distinguish anandamide, which is a very minor compound. In fact, Rafi Mechoulam always jokes about that he could only discover anandamide because he was probably using a pig brain that had been laying in the slaughterhouse for hours, and this was later shown to be a process that increases the levels of anandamide in tissues. So at that time, when he discovered anandamide, there were no mass spectrometers sensitive enough to measure picomolar or femtomolar amounts of these compounds. Now we do, and we can distinguish anandamide from its brother, which has 20 carbon atoms but only 3 double bonds instead of 4, and maybe that difference is enough for that compound to hit another receptor rather than just CB1 and CB2. So clearly, we have these days the possibility of measuring with high sensitivity, high specificity, high rapidity as well these compounds in all tissues. We can measure the expression of the enzymes that make these compounds with even at the single cell level. We can measure the proteins that are made by this. But by the RNAs, through the RNAs of the various proteins and enzymes and receptors. So we can have a real-time, almost real-time picture of what's happening in a given tissue or in a given biological fluid in a given animal which is undergoing a physiological or a pathological condition. So, and then of course, all this data these days, these are also the days of big data, of artificial intelligence. So all this data can be put together. There is the new approaches of the systems biology. So all the networks can be made sense of. And there is a lot of space for mathematics, not just for analytical chemistry and molecular biology. So we can have a picture. And at the same time, we have very sophisticated ways to modify, if not one single mediator of the endocannabinoidome, because that would be very difficult to do, but at least one single receptor in a single cell for those endocannabinoidome mediators. And by modifying that and then, controlling, like in an on and off situation, we can then understand what the endocannabinoidome mediators and the endocannabinoid system is doing. This has been done to a large extent in the brain because that's where all the research started, but it's now being done also in peripheral organs. And this is where we really appreciate the importance of the endocannabinoidome and the endocannabinoid system. They're not brain systems, they're really pleiotropic. They're doing a lot of things in a lot of places. And it's difficult to understand because, of course, you have lots of molecules being produced and degraded at the same time, but we must imagine that if these molecules are doing something important, and we have evidence that they are, they must have a way of coordinating themselves and being coordinated by other mediators, by other systems, physiologically and pathologically.
Yeah, and since all of this activity is so localized, if let's say 5-10 years down the road, if some of these analytical technologies become more accessible to hospitals and things, worldwide, would someone essentially have to have a biopsy on a particular set of tissues or, particular organ, that sort of thing, in order to get some sense of what's going on with endocannabinoids or receptors and that sort of thing, since they're not being circulated? Is that really the only way to kind of do that?
Unfortunately, this is why we still need the animal studies. We've been this is one of the reasons why I've been working on less invasive and more ethically valid systems like invertebrates or zebrafish. But at the end of the day, you cannot do without the animal studies. Also because there is genetic manipulations that you can do only in animals, fortunately, and at least for now. and obviously, we've done lots of studies with biopsies. The blood, when we look at the blood, of course, we get very interesting information from the blood, from the plasma, from the CSF, now also from the saliva from the feces. But obviously we don't really know where what we measure comes from, and this is therefore we can make correlations, we can make hypotheses, but we cannot really have a mechanistic link between what we see and the pathology or the physiological situation. Biopsies offer a better way, but then you have to be very, of course, you cannot always take biopsies from humans, certainly not from the brain. And
Yeah.
I've been trying to convince students to do that, but I haven't managed.
Yeah. I mean, geez. Yeah.
But you have to be careful even with biopsies, because they need to be taken in the right way, kept and stored in the right way. So there are less invasive ways. There is imaging techniques that are emerging, like PET techniques to image receptors, enzymes. Now you see that for the endocannabinoid system, and that's really raising the possibility of doing studies in vivo, in real time, also in human beings, in human volunteers. So I see honestly no limit, if not the technological limits of the time, which are probably going to be surpassed tomorrow or the day after tomorrow. The only thing we need is the realization that this is an important topic to analyze, because when you have hundreds of mediators coming more or less from the same pathway, or having to do with the same pathway which originates from the endocannabinoid system, but simply because that was the one we discovered first, but eating different, completely different targets, then you when you realize that this is important, then you will find a way of addressing the experimental problems and challenges. So it's just a matter of having the right questions, and you can get answers, at least partial answers. As we said at the beginning, a lifetime will probably not be sufficient to understand all these what all these mediators are doing, and how do they interact with each other.
Yeah, and one of the most important things in the scientific process is finding better questions to ask than you necessarily started out with.
And always having the right hypothesis, because I'm very much, even though with the omics you go fishing a little bit. I'm still a believer in the hypothesis driven research. So it's nice to have an hypothesis, and then of course, if you're lucky, you can get an answer to your hypothesis, and even if the answer is negative, the secret is not getting too attached to your hypothesis.
Exactly. Yeah, being more interested in just trying to understand what's actually going on.
You need to have a hypothesis to start with.
Yeah, to have a focus. Yeah, absolutely. One thing I wanted to make sure to talk to you about, because I know it's a focus on a lot of your research right now, which is the connection between the endocannabinoidome and the gut microbiome. And this is once again something that when I spoke with Ethan, he kind of teased a little bit of some of the work that's going on now. But can you speak a little bit to, I mean, obviously research these days is showing how the gut microbiome, all the gut flora affects so many different things, and how the immune system and psychology and all these other things are interconnected with it. But what is your research focusing on, and what are you currently learning about those connections between the endocannabinoidome and the gut microbiome?
This is, as I said at the beginning, I like difficult things, and I was given
That's a big one.
I was given the opportunity 4 years ago to start on a new project, and I must say I was immediately attracted to it, because the gut microbiome, I saw it a little bit like the endocannabinoidome, because it's not really an external part to our body. It's something that has been selected through generations, passed on genetically, but not only genetically, from mothers to children, and it's there for to play an important function. Has been conserved. It's not been fought by the immune system, so it's clearly something that we need, and all animals that have microbiota need them. So ideally it's a wonderful symbiotic relationship with another organ there is several more cells, microbial cells in our body than our own eukaryotic cells. So clearly, this offers the opportunity, if you think of it, of expanding a lot the potentiality of our own body. There is thousands of genes more than the ones we have, lots of more proteins and small molecules. And obviously what the gut microbiota does to help us, first of all, it does it to help itself. But obviously in the process, they produce molecules that can be useful to our own functions by reinforcing our own molecules, our own signals, but also by creating something that we don't have. But the experience of the endocannabinoidome told me that you've never enough chemical signals to deal with the complexity of life, and especially when it comes to dealing with the environment and the environmental challenges. Having a few thousands genes more, actually quite more than a few thousands more, definitely helped us and many other animals to survive and to better respond to the changing environment. Our own body is itself an environment for these microbes who live in completely different conditions. Like for us would be living in the North Pole or on the equator. So it's really fascinating, and it's and then you start seeing all these papers on the gut microbiota and the gut microbiome again. Here the difference between the word has to do with the fact that the microbiota are the microbes. The microbiome is the microbes, is their genes, is their proteins, is their small molecules, and so on and so forth. It's a bit like the endocannabinoidome. So beyond the names, what I saw and what everybody can see is that the gut microbiome is potentially involved, not just in controlling the function of our gut, in helping us with metabolizing things, but also in behavior, in the immune system, and in most of our functions. It is a bit like the endocannabinoid system and the endocannabinoidome, if you think of it. But it was really Patrice Cani in a nice paper that was published, I think, before 2010, who put the two things together, the endocannabinoid system, the gut microbiome. Patrice Cani is a scientist, a brilliant scientist in Belgium, and he showed that some of the effects of intestinal dysbiosis, the perturbation of the gut microbiome, because like all things, also like the endocannabinoid system, also the gut microbiome is perturbed, and the perturbation known as dysbiosis produces some of its negative effects on metabolism through the endocannabinoid system and through the CB1 receptor. So he was really a pioneer in this field. And at that time, I looked at the paper. It was very interesting, very nice paper. He published a few more. But then I was given the opportunity to work on a more or less similar subject, and I honestly didn't want to lose the opportunity. Also because it came with very nice funding, and which we don't always have in Italy. Yeah. We almost never have in Italy, and so I took this opportunity. But it's very complicated because I'm not a microbiologist, and the microbiota is very complicated. Although it's not there by chance, there are rules, but every person has a different microbiota. And but again, every person has a different profile of endocannabinoidome mediators. So again, maybe there are things that these two systems do together in collaboration, or one through the other. And there were many evidences that there would be a triangle between the endocannabinoidome, the gut microbiome, and mammalian physiology and pathology. So initially, we're still focusing on the metabolic, on obesity and metabolic disorders, which are obviously the first place to look for when you're talking about the gut microbiome, but also when you're talking about the endocannabinoid system. And what we're doing now is trying to prove that these two systems communicate.
Gotcha.
And again, there are things, there are relatively easy things that we could do. For example, use animals which don't have which don't have a microbiota at all. So they were raised in sterile conditions, and so we went and looked at their endocannabinoid system, their endocannabinoidome, and it's completely different. And this could be due to many. I mean, that doesn't really create a cause-effect relationship. But then when we reinstated the gut microbiome in these germ-free animals, in these germ-free mice, it took only 1 week to see almost, at least in the gut, almost exactly the same situation as conventionally raised mice. So that was really the evidence we were looking. That was the first experiment we did. That was really the experiment we did to say, okay, we stop here or we go. we go further.
Is it worth moving forward?
We decided to go further. Okay. And then, there are other things you can do, like knocking down enzymes of the endocannabinoid system, specifically in different parts of the body or in the gut, and see if the endocannabinoid system is modified. And again, we find changes. If the gut microbiome is modified, and we found changes. But it's a complicated field because, like with the endocannabinoidome, almost everything can change the gut microbiome.
Yeah.
And there is still no exact definition of what is a healthy or a pathological gut microbiome, exactly like the endocannabinoid system. So, and possibly also many other endogenous systems that we have. So there are many experiments we're doing. We find very interesting correlations in human studies following interventions between changes in the endocannabinoidome and changes in the gut microbiome. We find that if we modify the gut microbiome with probiotics or prebiotics or antibiotics, we see strong changes in the endocannabinoid system. We're now investigating whether the changes we see in the endocannabinoid system when we modify the gut microbiome are partly responsible for the effects of the modification of the gut microbiome. And the other way around, we try to see if when we change the endocannabinoidome in a specific manner, the changes we see in the gut microbiome participate in the effects of the changes of the endocannabinoidome. So we can do that using exactly the same tools we were mentioning before, plus more omics technologies like metagenomics to investigate the composition of the gut microbiome, and the proteomics to investigate their proteome. So, and eventually we will have to ask to mathematicians to look for algorithms that explain these correlations we find. But there are mechanistic ways to look at the relationship between these two worlds.
It's really fascinating, and the crosstalk is super interesting. That it's not a one-way street, that the endocannabinoid doesn't just affect the microbiome or vice versa, that it's back and forth. And I would imagine too, going back to something you mentioned very early on about the diet, and that we have certain requirements to have precursors for different things that our body needs to either construct the enzymes necessary to build up and break down endocannabinoids or receptors or whatever. So I imagine in just a simple manner, the microbiome would be involved in influencing how some of those those building blocks that we get from our foods are actually made available to the body to even use for some of those biosynthetic pathways.
Absolutely. There is such a large biodiversity in the gut microbiome that obviously you can imagine these bugs to do a lot of things.
Yeah.
They were discovered because we could see how some of these microorganisms helped us processing stuff that we're not able to process. What came out after that was that in the process of processing this, for example, complex fibers, which we cannot process because we don't have the enzyme to make them, these microorganisms, these bacteria, make molecules like the short chain fatty acids that are strongly active both on the gut but also on the brain, because they can travel in the bloodstream and reach the brain, or act on the peripheral nervous system and influence the behavior as well. So clearly, there are many possibilities that the microbiome can modify the endocannabinoidome. The microorganisms make enzymes that are similar to the enzymes that degrade the endocannabinoids. In fact, some of the commensal bacteria, they also make endocannabinoid-like mediators. So they make compounds, mediators that are very similar to the sisters and cousins of anandamide and 2,8-G. So and they were shown to act on the same receptors. So the chemical differences between these molecules when they're made by the microbes are not such to impede them to prevent them from interacting with the same receptors. So there is really a lot of possibilities for crosstalk. And of course, as you mentioned, even though we're perfectly capable of processing most fatty acids, there are fatty acids that we cannot make, like the polyunsaturated omega-3 and omega-6 fatty acids. So for that, we rely on the diet, and these fatty acids can change the function of the microbiome. So the omega-3 fatty acids found in fish oil, for example, we knew already that when you supplement them in the diet, they can change the endocannabinoidome. Now we know that they can also change the gut microbiome, and some of the changes we see in one or the other are probably due to the fact that they're changing the other rather than the former, so the latter rather than the former. So it's really interesting. We have what is really difficult here is to find models to study this interaction, because we cannot really do it in an in vivo setting. So we're developing in vitro systems, for example, the organoids, where we can keep pieces of gut alive for several months. We're now trying to find a way of inoculating them with gut bacteria without killing them, because normally organoids are grown in antibiotics. So we need to
makes it a little challenging.
Yes, it's a bit challenging. But we can at least do this in an indirect way. So we can take the media of the bacteria and see the effects that they have on the organoids. But we can also do experiments on the microbiome outside the organism, using the so-called simulator of the human intestinal microbiome ecosystem, or SHIME. So we have a SHIME in Quebec, where we basically instate a real human microbiome. So there is several flasks, each of them mimicking the conditions you find in the stomach, in the duodenum, in the ileum, in the colon. So and if you put a fecal sample of a human being in this system, after a couple of weeks, the bugs will start populating the different parts of the SHIME in a way similar to what they do in a human gastrointestinal system. And at that point, when we have this SHIME, we can give it foods, we can give it fish oil, for example, we can give it polyphenols, we can give it many dietary stuff, and see what happens independently of the host. And then we can find a way of introducing the host cells in this SHIME. And this is more complicated, but people are working on this. There are very strong groups in the Netherlands who are working in simulating the microbiome and the host interactions on a chip, for example, or in very small systems. So once again, technology is just waiting for us to ask the right questions.
Absolutely, yeah. That is so fascinating to learn about how that research is done. That's been something I've wondered for a while, exactly how you start to piece all that out, because it is so complicated. And it sounds like you and I both share an interest in complex systems. That's something that that I definitely like as well. I like puzzles, and so all of this is very fascinating. And I want to be sensitive to your time. I know we're we're creeping over over the hour here. I've got one last question. One thing that I think people misunderstand about the entourage effect is that in the research that was originally published, that you were an author on, that first described this effect, is you were looking at 2AG and you noticed that there were these other compounds that didn't seem to have much activity of their own on these receptors, but when they were present with 2AG, you got unique effects. Is it fair to say that the concept of the entourage effect has been a little manipulated in popular culture to sort of take on a meaning that is different than what was originally intended?
Yeah, absolutely. I agree. And both Rafi and I wondered sometimes whether or not the entourage idea had not been kind of misinterpreted or misunderstood in many cases, because it has been applied to cannabis and to the pharmacology of cannabis, which is something that for a pharmacologist has really no meaning, because you cannot speak about the pharmacology of a plant. You can speak of the pharmacology of the components, of the chemical components of the plant, and even that is quite difficult to do. So clearly, the entourage effect, as we conceived it, was born to explain why anandamide and 2AG in particular was accompanied by, in tissues, in blood, in wherever we analyze it, by congeners, by other monoacylglycerols, which at that time were thought to have no receptor. So they were orphan of receptors. Okay, so we wonder, why would the cell make 2AG together with these compounds, or anandamide together with its congeners, if these congeners have no biological action? That would be a waste of a biochemical machinery. And then, and he said, okay, maybe they are doing something to the main actor of the scene. And I thought that something could be to retard their degradation, because as I mentioned, both the N-acetylethanolamides and the monoacylglycerols can be degraded by the same enzymatic pathways as anandamide and 2AG. So clearly, they can compete for their degradation by this enzyme, and through this means they can they can prolong their lifespan, and then enhance their activity. And this is what we found, and this is what we demonstrated in that paper. But then the concept went on forward, and since for some of these compounds it was really difficult at the beginning to find a receptor, we carried out other different kind of experiments. For example, whether these compounds could act as allosteric modulators of the receptors, and in fact we found that one of them, palmitoylethanolamide, later on we showed would enhance the activity of anandamide at 3B1 channels. So these were really the early times of endocannabinoid research, and then many groups, including the group of Daniele Piomelli and others, showed that in fact these congeners were not inactive. They had their own receptors, and these receptors in some cases did things quite different from the CB1 and CB2, and in some cases things even opposite. So the entourage effect was kind of crushed by this idea, because if 2AG acts on CB1, and by acting on CB1 stimulates food intake, stimulates fat accumulation, and its congener act on another receptor known as GPR119, which is doing exactly the opposite, there is no more entourage effect at the organismal level. So that was really something to be interpreted for the particular historical situation, and I think it's still valid in some cells. For example, because in all cells these compounds are produced together with their congeners, and not necessarily the receptors of these congeners are there. So when they're not there, maybe, and when CB1 and CB2 receptors are there, maybe they're actually acting as entourage compounds. The situation in the plant, where it has been proposed that some terpenoids could enhance the activity of THC or CBD, or that other phytocannabinoids could enhance the activity of THC, CBD, is completely different. And I don't know if there is an entourage effect in cannabis. I don't know whether it explains anything, if there is one. but clearly it's different from what we conceived in the 1990s with Rafi.
Yeah, absolutely. And I think that's a really big thing that a lot of people listening to this are unaware of, mostly because, I mean, a lot of casual, lay people that are interested in cannabis, a lot of them aren't going to the original research papers and reading the technical details on how these things came about. So I think that's very important to point out, because a lot of times what I teach is that we now know enough to realize how much we don't know. And, and we have learned a lot. But
I
a
think,
lot of times in the industry, a
yeah,
lot of claims are made
yeah.
that we know a lot more than we
I
actually
think
do.
the entourage effect, as it is being used today, is something different. It's basically stating another potential truth: is that the effect of the sums of the effect. Okay, the effect of the single components together is stronger than the sums of the effects taken alone. Okay,
Right. Yes.
This could be true, but it's not the entourage effect that Rafi and I thought about. Okay, the entourage effect was something that comes together with the main actor and is protecting it from being. It's a real entourage effect if you think of it. From being degraded, from it's enhancing its activity, its visibility, and the entourage, the synergistic effect that you find in some plant preparations. It's probably, I'm not saying it's not true, but it's different from what we found, and it's probably due to different components acting on different receptors in a synergistic manner, rather than in an additive or counteractive manner.
Yeah, absolutely. The way I always describe the entourage effect, because I'm always kind of I'm a hyper analytical person anyway, but I always say that the entourage effect was initially conceived to describe an effect that has components that generally you couldn't identify their effects by themselves, and then in together they had unique effects, which is slightly different than this sort of herbalism synergy kind of concept. Yeah, so that's really great to clarify. Well, before any more hurricanes come your way and knock us out here.
I hope it's enough for today.
I know, yeah. Off recording, we were talking about how the storms everywhere are just getting so bad, and we've been struggling to finish the interview because of thunderstorms coming through.
But you can put things together, right?
Oh yeah, I'll edit it together. No one will know the difference. But Dr. Di Marzo, thanks so much for being willing to spend. I know I took up about 30 minutes more of your time than we planned, but
it was not our fault.
Yeah. Well, I really, really appreciate it, and I'm going to be following your work as things continue for you.
Thank you very much. It's very, very nice. It was nice speaking to you. Very, very nice questions.
Yes, thank you. Yeah, I'm glad our paths could cross. And do you mind if in the future I reach out to you again sometime, maybe next year or so?
No problem.
Excellent.
I hope to be able to tell you something interesting regarding the interaction with the gut microbiome.
Absolutely. If something comes up that gets you excited, feel free to email me, because I get, I'm excited about all this stuff too. So thanks so much for sharing your insights with our listeners. Take it easy and have a great rest of your day and week.
Thank you very much. And you too. Take care and stay safe.
If you want to learn more about cannabis, check out the Curious About Cannabis book on Amazon.com and other major online book retailers. To support the show and get access to an exclusive members-only podcast feed, access to private events, merchandise discounts, and more, visit www.patreon.com/curiousaboutcannabis.